Imagine you order like a highly anticipated, ridiculously expensive package.
Yeah.
Maybe it's this fragile, cutting-edge piece of new technology
Mm-hmm.
that you've been saving up for, and you've been waiting months to get it.
Right.
And you finally get that notification on your phone, the one that says, out for delivery.
The best feeling.
Exactly. You're thrilled. But here's the problem. Every single time the delivery driver gets anywhere near your house, this group of hypervigilant bouncers intercepts the package and just throws it straight into a wood chipper.
Wow. That's a brutally frustrating scenario because you paid for the delivery, the driver navigated the entire route perfectly, but the package never even makes it to your front door because of this overwhelming, just aggressive security force standing in the yard.
Well, welcome to the Deep Dive Lab. Because you are that delivery driver, and in the real world, that package is like a billion-dollar cancer drug or revolutionary gene therapy.
Exactly.
Today, we are investigating one of the most expensive and honestly confounding mysteries in modern medicine. Like, why do some of our most advanced, life-saving nanomedicines simply disappear in the body before they ever reach their targets?
That's a huge problem.
And the crazy part is, the answer scientists have finally uncovered involves this bizarre connection between your stomach, a mood-altering chemical, and those resident bouncers throwing your medicine into the wood chipper.
Yeah, and the path to solving this mystery is honestly just a masterclass in shifting perspective.
How so?
Well, for decades, the scientific community was completely stuck, basically looking at the wrong part of the body. But to understand how they finally broke through, we first need to look at the delivery vehicles themselves.
Okay.
In medicine, we call these in vivo delivery systems, or IDS.
Right, so give me a tangible example of an IDS, like something people might actually interact with in a hospital clinic.
Sure. The most famous example today is probably the lipid nanoparticle, or LNP.
Okay, the mRNA vaccine stuff.
Yeah. Exactly. It's this microscopic fat bubble used to deliver mRNA in those recent vaccines. Because if you just inject raw genetic code directly into the human bloodstream, the body's natural enzymes will they'll chew it to pieces in a matter of seconds.
It's super fragile.
Incredibly fragile. So scientists engineered these tiny lipid armored cars to protect the mRNA, shuttle it through the sort of chaotic environment of the blood, and safely drop it off inside a specific cell.
And it's not just vaccines either, right? I mean, we're using these systems to deliver CRISPR gene editing tools or synthetic polymers loaded with highly toxic chemotherapy designed to strike a tumor and literally nothing else.
Yeah, that is the absolute dream scenario. You load the fragile or toxic cargo into the vehicle, you inject it, and it just drives straight to the destination. But that brings us right back to your wood chipper analogy.
The bouncers.
Yeah. The dark secret of nanomedicine is a staggering failure rate. Retrospective studies examining these advanced delivery systems reveal that less than 5% of them actually reach their intended targets.
Wait, less than 5%?
Less than 5%.
That means 95% of these incredibly expensive, highly sophisticated therapies are just vanishing into the ether. Like, where's the body even sending them?
They are being rapidly scavenged and destroyed by the liver.
The liver.
Specifically, by a specialized group of resident immune cells called Kupffer cells. Kupffer cells are a type of macrophage, which is a term that literally translates from Greek as big eaters.
Big eaters.
Yeah, they just line the blood vessels of the liver, acting as this massive biological filter.
So they're the bouncers in our yard.
They are the ultimate bouncers. Their entire biological purpose is to constantly sample the blood rushing past them, just grabbing and destroying anything they don't immediately recognize as a native part of the body.
And they're good at it.
Exceptionally good. They look at these billion-dollar lipid nanoparticles and viral vectors, flag them as foreign invaders, and simply devour them.
So if I'm a pharmaceutical scientist, right, and my targeted cancer-curing nanomedicine is getting eaten by the liver before it even gets close to the tumor, I feel like my first instinct would be to just make the medicine harder to see.
Right.
Like, I'd want to put better camouflage on the delivery driver so the bouncers just don't notice him walking past.
And you are tracking perfectly with the last 20 years of material science.
Oh, really?
That is exactly the instinct the entire industry followed. I mean, they spent billions of dollars and decades of research trying to make the delivery vehicles stealthier.
Did it work?
Well, they developed complex chemical coatings, like this process called pegylation, to create what they termed anti-fouling surfaces. They endlessly tweaked the lipid structures, hoping to build the perfect disguise to just slip past the liver.
But I'm guessing from the tone of this investigation that the whole camouflage strategy didn't really pan out.
Yeah, unfortunately. I mean, it achieved some minor incremental successes, but it never solved the core problem.
The bouncers were just too good.
The Kupffer cells were always too vigilant. The liver remained an absolute black hole for nanomedicine. The disguises just weren't enough to overcome the baseline aggression of those cells.
Which means the entire field was fundamentally asking the wrong question.
Exactly.
Instead of constantly trying to build a better disguise for the delivery guy, someone finally needed to stop and ask, why are the bouncers so aggressive in the first place?
Yes, that was the massive blind spot. Nobody was asking why the liver maintains this incredibly high baseline capacity to just eat everything in sight.
Okay, so who finally asked the right question?
That brings us to the real breakthrough. A groundbreaking study published in March 2026 in the journal Science.
The big one.
Yeah. It was led by Dr. Qin Wang, along with Wei Jiang, Xu Zhu, Yukai Wang, and their team of researchers at the University of Science and Technology of China, and they decided to look entirely outside the liver to solve this liver problem.
Wait, where did they look?
They looked at the gut.
The gut.
Yeah. Biologists have known for a long time that there's this intimate crosstalk between the gut microbiome, the trillions of bacteria living in our intestines, and the immune system.
Right.
Anatomically, there is a major blood vessel called the portal vein that connects the gut directly to the liver. So Dr. Wang's team formed this wild hypothesis.
What was it?
What if the bacteria in our stomach are somehow sending signals that keep the liver's Kupffer cells in a constant state of high alert?
Okay, testing that hypothesis seems like a complete logistical nightmare. I mean, how do you even isolate the behavior of gut bacteria to see if they're telling the liver to eat cancer drugs?
Well, you start by turning off the bacteria.
Oh.
The researchers ran a really elegant foundational experiment. They took mice that had various types of advanced tumors, so colon cancer, breast cancer, melanoma, and they administered a broad-spectrum antibiotic cocktail for a few weeks.
Just wipe them out.
The goal was to completely wipe out their gut microbiomes. Once the gut was essentially sterile, they injected those mice with different delivery systems, including targeted chemotherapy and oncolytic viruses designed to attack the tumors.
Okay, wait, hold on a second. We are constantly told by every nutritionist and doctor how absolutely vital our gut microbiome is for our overall health, our digestion, our immune system.
Right.
Aren't you basically nuking the patient's entire biological ecosystem just to deliver this drug? That sounds incredibly dangerous.
Yeah, that is a crucial point to clarify. Administering broad-spectrum antibiotics to wipe out the microbiome is absolutely not a viable long-term treatment for a human patient.
Okay, good.
You're completely right. The collateral damage to digestion and immunity would be severe. The researchers were not proposing this as the cure. This was strictly a proof-of-concept experiment to see if the gut bacteria were actually the ones holding the strings. They just needed to isolate the variable.
Understood. So in this controlled proof-of-concept environment with the gut bacteria completely out of the picture, what did the delivery vehicles do?
Oh, the delivery rates just skyrocketed.
Really?
Yeah. Without the gut bacteria present, we saw an immediate doubling of tumor accumulation for synthetic nanoparticles and a threefold increase for the viral vectors.
Wow.
The cancer drugs actually reached their destinations. The tumors shrank dramatically. In some of the mice with colon cancer, this intervention allowed the targeted chemo to completely halt tumor growth, massively extending their survival.
That is a staggering difference. Did this apply to the genetic therapies as well, or was it just the cancer drugs?
No, it applied across the board. When they tested mRNA gene editing, the efficiency of the editing jumped 5 to 15-fold in organs outside the liver, like the lungs and the bone marrow.
So the Kupffer cells essentially just fell asleep on the job the exact moment the gut bacteria disappeared.
Pretty much.
Did the researchers physically see this happening?
They did. They used high-resolution intravital microscopy to observe the Kupffer cells inside living mice.
And what did it look like?
Well, normally, a Kupffer cell looks a bit like a microscopic octopus. It has these long, dynamic, tentacle-like structures called pseudopodia.
Tentacles.
Yeah, and they use these tentacles to physically reach out into the rushing bloodstream and snatch passing nanoparticles.
Kind of like reaching out and grabbing our delivery driver by the collar.
Precisely. But in the mice without gut bacteria, the physical structure of the Kupffer cells fundamentally changed.
How so?
They lost their tentacles.
No way.
They shrank down, became rounded, and turned into these smooth, compact little spheres. They were functionally deactivated. The nanomedicines just floated right past them, completely ignored.
Okay, this mechanism is absolutely fascinating, but it introduces a major logistical gap.
What's that?
The bacteria in our colon are not physically swimming up the portal vein to hand out tentacles to the liver cells. There has to be some kind of messenger carrying the instructions from the gut to the liver.
And that is the exact puzzle the researchers systematically tracked down next.
So what did they find?
They discovered that the bacteria were not sending their own molecules directly to the Kupffer cells at all. Our own bodies were acting as the middleman.
Oh, really?
Yeah. The lining of your intestines is made up of this sensory network of cells. Think of them as tripwires.
Okay.
When these intestinal epithelial cells detect the presence of specific gut bacteria, an alarm goes off. That alarm triggers the intestinal cells to release a chemical messenger right into the bloodstream, which travels straight to the liver.
What is the messenger?
It's serotonin.
Wait, serotonin.
Yes.
The famous happy chemical in our brains that regulates our mood and sleep. You're telling me that serotonin is moonlighting as a trigger that turns liver cells into microscopic, tentacled monsters that eat our medicine.
I know it sounds completely like science fiction, but yes.
That's wild.
We culturally associate serotonin almost entirely with the brain and our emotions, but over 90% of the body's serotonin is actually produced and stored in the gut.
90%.
Yeah. And the researchers found that when the intestinal tripwires sense the gut bacteria, they ramp up the absorption of tryptophan,
Tryptophan, right?
which is the raw amino acid material the body uses to synthesize serotonin. The gut rapidly manufactures serotonin and pumps it directly into the blood, heading for the liver.
Okay, let's trace that signal because this is crazy. The wave of gut-derived serotonin hits the liver. How does a mood chemical force a liver macrophage to grow tentacles?
So the Kupffer cells in the liver have specific docking stations on their surface built exclusively for serotonin, receptors called HTR2C and HTR7.
Okay.
When the serotonin binds to these docking stations, it acts like a massive cellular power-up. It triggers this process called cytoskeletal remodeling.
What do you mean?
The internal physical skeleton of the cell rapidly assembles dense networks of actin filaments, which literally push the cell membrane outward, sprouting those grabbing tentacles.
It's instantly arming them.
Exactly. And simultaneously, the serotonin forces the Kupffer cells to upregulate their eating genes, meaning they express far more receptors designed to recognize and consume foreign objects.
From an evolutionary standpoint, why on earth would our gut bacteria want to give our liver cells tentacles? Like, what is in it for them? It feels almost like a design flaw that our own microbiome is sabotaging our medicine.
To understand the evolutionary why, you kind of have to look at the human digestive tract for what it really is. A long tube of the outside world running directly through the middle of our bodies.
That is a gross but accurate way to put it.
It's loaded with trillions of bacteria, and it's just a messy, chaotic environment. Sometimes those bacteria break through the intestinal wall and enter the bloodstream.
And the blood from the gut goes straight to the liver.
Exactly. The liver is the very first checkpoint. It is the fortress guarding the rest of the body from blood-borne gut pathogens. So over millions of years of evolution, the gut and the liver developed this brilliant communication system.
A threat level indicator.
Yes. The gut essentially uses serotonin to send a threat level signal to the liver. It says, hey, we have a very high bacterial load down here today. Keep those tentacles out and be ready to eat anything that slips through.
I see the logic now. So the nanomedicines are not the intended target at all.
Not at all.
They're basically just catching stray bullets in an ancient ongoing border war between the gut and the liver.
That is a perfect synthesis. The lipid nanoparticles and viral vectors just happen to look like the tiny foreign invaders the liver has been trained by the gut to destroy for millions of years. It's a continuous, steady-state feedback loop that keeps your liver's immune defenses dialed up to maximum capacity.
Well, this realization entirely reframes the problem. We don't need to try and out-engineer millions of years of evolutionary defense mechanisms with better camouflage.
Right.
If gut-derived serotonin is the power-up keeping the bouncers aggressive, we just need to temporarily turn off the power.
Which brings us to the application phase of the study, where this goes from, a fascinating biological quirk to a literal medical revolution.
Yes, let's get into this.
The researchers proved that by briefly interrupting the serotonin pathway, you can safely sneak medicines past the liver. And they demonstrated this using two entirely different, highly practical methods that do not involve nuking the microbiome with antibiotics.
Let's hear the first method. How do you turn off the serotonin supply without harming the patient?
The first approach was a dietary intervention. I mentioned earlier that serotonin is synthesized from tryptophan, right?
Yes.
Tryptophan is an essential amino acid, meaning the human body cannot manufacture it from scratch. You can only acquire it through the food you eat, typically protein-rich foods.
Like everyone always blames the tryptophan in the Thanksgiving turkey for making them sleepy.
Precisely the same molecule. So the researchers took mice with breast cancer and colon cancer and put them on a strictly tryptophan-free diet for just 5 days.
Only 5 days.
Just 5 days. Because the body can't make its own tryptophan, the raw materials ran out quickly, the serotonin levels in the blood plummeted, and without that constant serotonin signal arriving from the gut, the Kupffer cells in the liver lost their tentacles and reverted to those smooth, dormant spheres.
And once the bouncers were deactivated, they sent in the delivery drivers.
They injected the lipid nanoparticles and the gene editing therapies, and the delivery to the breast and colon tumors literally doubled.
That's amazing.
The protein replacement therapies became wildly successful, shrinking the tumors down dramatically.
I could see a flaw here, though.
Oh.
I mean, you can't just leave a sick cancer patient on a heavily restricted tryptophan-free diet indefinitely. Amino acids are, well, they're essential for maintaining muscle mass and overall cellular health.
You are spot on, and that is the beauty of this dietary hack. It is entirely temporary and reversible.
Oh, really?
Yeah, they only needed the diet to last long enough to open a window of vulnerability. Once the nanomedicine was safely delivered past the liver and into the tumor, they put the mice back on a normal, healthy diet.
Oh, wow.
Their tryptophan levels replenished, the serotonin bounced back, the Kupffer cells grew their tentacles again, and the liver returned to its normal defensive state. You open the window, drop the package, and close the window.
That is remarkably elegant. But I imagine asking a patient undergoing severe chemotherapy to radically change their diet, even for 5 days, could be challenging. What was the second method the researchers developed?
The second method was pharmacological, and it's arguably even more exciting because it relies on existing infrastructure.
How so?
Because serotonin is such a critical neurotransmitter in the brain, pharmaceutical companies have spent decades developing hundreds of drugs that target specific serotonin receptors to treat neurological disorders like anxiety, depression, and migraines.
So they didn't even have to invent a new drug to solve the nanomedicine problem.
Not at all. The researchers selected two existing drugs, ritanserin and AVN-101. These are serotonin receptor antagonists.
So instead of fighting the liver cells head-on or putting a better disguise on the delivery driver, these drugs are acting like a radio jammer.
A radio jammer, yeah.
The gut is still pumping out serotonin, screaming over the radio for the liver to attack, but the drugs block the docking stations. The liver just hears static.
I love that radio jammer analogy. That is exactly the mechanism. The drugs sit in the keyhole so the serotonin cannot enter. They created an antagonist cocktail of these two existing drugs and administered it to the mice for just 1 week before injecting the cancer therapies.
And the microbiome was left completely intact.
Completely intact. Digestion and gut health were unaffected. But the Kupffer cells were completely deaf to the serotonin signal.
And results.
The results were phenomenal. This radio jammer cocktail safely allowed chemotherapy and oncolytic viruses to bypass the liver and reach the tumors at incredibly high rates. It significantly extended the survival of the mice with aggressive melanoma. And the most important part, they achieved this without having to alter the actual cancer drugs in any way.
This feels like a massive paradigm shift.
Yeah.
I mean, we're talking about billions of dollars poured into material science, trying to build stealthier nanoparticles from scratch, when the answer was just a brief diet change or a temporary dose of an anxiety medication.
It radically shifts how we approach therapeutic delivery. If we zoom out and look at what this means for the future of medicine, there are massive implications.
Like what?
First, we now recognize that the liver's aggressive immune clearance is not an isolated function. It's actively puppeteered by the gut. Second, by temporarily hacking that connection, we can vastly improve the efficacy of the mRNA vaccines, the CRISPR tools, and the targeted therapies we already possess.
Right.
We don't have to wait 20 years to invent a new invisible armored truck. We just pave a clear road for the existing trucks to do their jobs.
You know, there's another angle to this that we haven't touched on. If 95% of a highly toxic chemotherapy nanodrug is getting eaten by the liver instead of reaching the tumor, that must cause massive collateral damage to the liver itself.
It absolutely does. Liver toxicity is one of the most severe limiting side effects of these advanced treatments. It restricts the dosages doctors can safely give.
Because we just destroy the liver otherwise.
Exactly. So by using a serotonin blocker to turn off the Kupffer cells, you are not just getting more medicine to the tumor, you're simultaneously rescuing the liver from being poisoned by the very drugs meant to save the patient's life.
Wow.
You increase the efficacy and massively decrease the toxicity in one move.
It is genuinely incredible. We spend so much time in modern medicine treating the human body like a machine with isolated modular parts.
So true.
If you have a liver problem, the instinct is to only look at the liver. But here we have an ancient evolutionary web. Bacteria living in our intestines are communicating via a mood chemical to dictate the immune response of our liver.
Yeah.
And this ancient web is ultimately dictating the success or failure of cutting-edge 21st century synthetic medicine.
It reinforces the reality that you cannot engineer a biological solution without deeply understanding the ecosystem you're deploying it into. The gut-liver axis was a blind spot that defeated nanomedicine for 2 decades. By simply understanding the conversation happening inside the body, scientists turned that blind spot into a powerful therapeutic tool.
We've spent this entire deep dive focused on medicine and drug delivery. But if the microscopic bacteria in our stomach have the power to use a mood chemical to physically alter the cellular skeleton of our liver, it really makes you wonder about the broader implications of that signaling network.
In what sense?
Well, we culturally think of our mood, our energy levels, and our personality as things that belong uniquely to us.
Right.
But if our microbiome is constantly pumping out 90% of our serotonin to direct cellular traffic in our internal organs, you have to wonder how much of you is actually you, and how much of your daily experience is just a chemical text message sent by the trillions of microscopic passengers living in your gut.
That is a profound implication. We are just beginning to map the chemical language of the microbiome, and we're finding its influence in places we never expected to look.
Something for all of us to think about the next time we crave a turkey sandwich. Thank you for joining us today in the Deep Dive Lab. We hope you enjoyed uncovering the hidden mechanisms of the gut-liver axis as much as we did. If you want to keep exploring the unexpected connections that drive our world, make sure to follow and subscribe for more science stories. We will see you on the next Deep Dive.